Hello, and welcome everyone to this week's seminar. So just to say again, we are still accepting offers for talks from both early career researchers or from group leaders. So the links are in the description of the video. If you'd like to submit a talk, we would love to hear from you. So I will just introduce our first speaker for today. So we have Audam Chhun from Warwick University. So if you want to just go ahead and take it away, I will mute myself.
Thank you. Is that okay now? Hi everyone, thank you for attending this seminar, and thanks for the organizers to organize this. So my name is Audam. I'm a final year PhD student in Chris Core and Joseph Christopher's group at the University of Warwick in the UK. And I'm going to present a bit of my PhD work, which focused on the marine actinobacteria Salinispora tropica for the study of novel natural products. So before talking about marine bacteria, I just want to briefly mention very important bugs that are in the soil. So in the soil, we have Pseudonocardia, which are part of the Actinomycete family. And they're a very important bug for us because they produce a lot of bioactive compounds, such as antibiotics. And they produce so much so that up to 70% of all the antibiotics currently used in the clinic come from this genus Pseudonocardia. But because it's been extensively studied over the years, we've seen a decline in the rates of discovery of new compounds from Pseudonocardia. And this is when people started to have an interest in actinomycetes in completely different environments, such as the ocean. The rationale being that if we found actinomycetes in different environments, they may produce completely different compounds as well. So a few years ago, marine actinomycetes were discovered in marine sediments, and it was called Salinispora. Sequencing of its genome a few years later revealed the presence of 20 biosynthetic gene clusters, which are colocalized genes that code for most natural products. So one example of a new molecule that was discovered in Salinispora is this compound called salinisporamide, which is an anti-cancer molecule that is currently tested in the clinic. So salinispora has proven itself to be a promising and valuable strain for the discovery of novel natural compounds. But what's even more interesting is that if you look again at the biosynthetic gene clusters that were found in Salinispora, so here in orange I depicted the characterized gene clusters. So it's the gene clusters for which we know the product. And I've also showed in green the orphan gene clusters, which are the gene clusters for which we don't know the product. So another way to look at this is that up to 60% of Salinispora's biosynthetic potential is being locked away. There's still many orphan gene clusters that still remain uncharacterized. So one way to try to activate these silent gene clusters is by co-culturing. As you can imagine, that certain bug will require competition or just interaction with other microbes to express certain pathway and express certain molecules. So a few years ago, a group in the US co-cultured Salinispora with different marine heterotrophs. And they saw that Salinispora was actively killing these heterotrophs, but they've been unable to identify the killing agent in these conditions. So what I've been doing for my PhD is to actually co-culture Salinispora with marine phototroph, and especially these cyanobacteria called Synechococcus. And we also observed in co-culture that Salinispora was able to kill our cyanobacteria. So what we think happened is that Synechococcus released something in the media that activated a previously silent gene cluster in Salinispora. And part of my PhD work was to try to understand this mechanism and identify the potential novel antibiotic being expressed here. So as I just said, we've studied this cyanobacteria, Synechococcus, which has a red pigment for photosynthesis. So here you have pictures of actinic cultures of Synechococcus over time. So you can see it's red, and with time it becomes more red and thicker because the cells are healthy. But as soon as you co-culture it with Salinispora, which is the bottom line, you can see cell bleaching, which indicate death of the phototroph, indicating that Salinispora has negative effect against it. Another way to look at this is by flow cytometry. So Synechococcus autofluoresce, so it's quite easy to measure the cell abundance. So here, this graph shows the phototroph cell abundance over time. In green, it shows the measurement of an actinic culture of the phototroph, which is our control. And in blue, you have the measurement of the phototroph in co-culture with Salinispora. And again, you can see that as soon as we put our phototroph with Salinispora, we observe a steady decline in cell number. So Salinispora is killing our phototroph. We then wondered if this killing phenotype could also be applied to different type of phototrophs. So we've repeated the co-culture. So here again, you have phototroph abundance over time. So the red is still our control, which is just the phototroph going by itself, and in blue is the co-culture. So on the left, you have, again, our co-culture with our cyanobacteria, Synechococcus. And what was interesting is that we've also co-cultured Emiliana huxley, which is a coccolithophore. And we can also see that Salinispora was able to kill the entire population of Emiliana huxley. And we've also tried to co-culture Salinispora with Phytodactylum, which is a diatom. And it's less dramatic, but you can see that there's a difference in cell number between the actinic conditions and the co-culture. So what was very interesting about these results was that, so Synechococcus is a phytoplankton, it's cyanobacteria, so it's prokaryotic cells, but Emiliana huxley and Phytodactylums are actually eukaryotic cells. So it was quite interesting for us to see that the antimicrobial effect that Salinispora has works against both eukaryotic and prokaryotic phototrophs. So we knew it was able to kill phototrophs, and then we wondered if this killing was happening through an actual antibiotic molecule or through self-cell interactions, for example. So typically we've done our co-cultures in flasks. So in red here, you have again our control, which is just the phototroph going by itself. And in blue, you have the co-culture with Salinispora. So the way to test this is that we've set up our co-cultures in these small wells where the cells are physically separated. So for example, here for the co-cultures, you can see that we put Salinispora in the upper compartments, Synechococcus in the lower compartments. And between them, they're separated by a porous membrane that allows a small molecule to go through, but the cells don't physically interact. So using this setup, we've been able to show that Salinispora was killing our phototroph through the release of the molecules, because you can see here that in the co-culture, even with the inserts where the cells don't physically interact, Synechococcus was still killed by Salinispora. So we knew Salinispora was able to kill different type of phototroph, and it was happening through a small molecule. So our next logical step in the project was to try to identify this molecule. So to do this, we have analyzed the supernatant of either the phototroph going by itself, which is in red, or of our co-culture supernatant in blue by non-targeted metabolomics. And we've been able to identify 8 molecules, that were detected only in co-cultures. So then we worked towards trying to understand what these molecules were. So for example, if you look at molecule 1, 2, 5, and 8 here in orange, we've been able to partially characterize them by high resolution mass spectrometry to get their exact mass and empirical chemical formula. And through different type of analysis, we've been able to assign these molecules to 2 analogs of salinisporamide, which is again the anti-cancer molecule that I mentioned before. So for example, we've been able to show that salinisporamide A can get hydrolyzed and form the compound number 5. And it can also react with tris, which is a buffer we use in our media to form compound number 1. And similarly, salinisporamide B can also get hydrolyzed and form compound number 8 and react with tris to lead to compound number 2. So basically, these 4 compounds are all related, and they're all derivative products of 2 analogs of salinisporamide. So these compounds have been previously characterized, but because we're seeing so much of this in the culture, we wanted to know if it had antimicrobial activity. So we obtained 2 mutant strains from a group in the US. So the first mutant was SAL-L that didn't produce salinisporamide A. And we've also had SAL-A that does not produce neither of these analogs of salinisporamide. So we then use these mutants to repeat the co-culture experiments. So here again, you have the phototroph cell count over time. In red, you have the control, which is just the phototroph growing by itself. In green and in blue, you have the co-culture with the 2 mutant strain of Salinispora, and in purple, you have the co-culture with the wild-type strain of Salinispora. And basically, this graph shows that after 35 days, both mutants, even though they don't produce these compounds, they were still able to kill a phototroph, indicating that these compounds were not involved in the killing of the phototroph. So we then focused on these 3 molecules in green, so 4, 6, and 7. And again, by high resolution mass spectrometry, we've characterized them by getting their exact mass and empirical chemical formula. And what you can see is that these compounds are related. They share a similar backbone. So for example, between molecule 4 and 6, you only have the addition of 1 hydrogen and the removal of 1 nitrogen and oxygen atom. And between molecule 6 and 7, there's basically just 1 methyl group difference. So what was really exciting for us is that then with this information, we look for these molecules in different natural product databases, and we didn't get any match, which indicated that these molecules were new and they've never been reported before in the literature, which was super interesting. So then our next step was to try to characterize them further and to get to their structures using NMR. So to do this, you need to purify your compounds in high amounts. So I've developed an LHPSC method to try to purify my compounds where I used a grain of methanols, and I've then collected different fractions here. And then by LCMS, you can check this fraction and see where your compound is. So for example, here I've been able to show that using my methods, if you look at fraction number 5, I've been able to purify one of my compound of mass 435, which is my compound number 6. And I've been able to isolate it in pure 4 from the other molecules. So I have improved and tweaked this protocol, and I've applied it to up to 4 liters of co-cultures to try to get a lot of these compounds for NMR, but unfortunately, after several attempts, it didn't work. It is quite tricky to increase the volume of these co-cultures, and we think there was just not enough of the compound. It's produced in very low amounts, so it's hard to get enough of it for NMR. But even though we haven't managed to characterize the structure of these molecules, we wanted to characterize them further. So first, we wanted to check that they were also produced with other phototrophs that we tested before. And as you can see, we were able to detect these compounds with when Salinispora was co-cultured with the other phototroph, which was good. And what was very interesting is that we've realized that these compounds the production of these compounds were actually triggered by the presence of the phototroph. So here is again LCMS analysis from the supernatant of Salinispora growing axenically in marine broth, which is a very rich media in which Salinispora grows well and is very active. And then we've also analyzed the co-culture, the supernatant of our co-culture. And you can see here that our 3 molecules of interest are only produced and detected when the phototroph is present and not when Salinispora is growing by itself. And another way to look at this is that we've, again, analyzed the supernatant of our co-cultures, which is in red. And we've also inoculated Salinispora in the supernatant of our phototroph. So we grew our phototroph for a while, then we removed the cells, we inoculated Salinispora into it. So basically there is no phototrophic cells, but there are all the nutrients and molecules that they release. In green, and in black, we've inoculated Salinispora in artificial seawater, which is the media we use for our co-cultures as a control. And you can see that the molecules were detected in co-culture, which makes sense, but also when Salinispora was growing in the supernatant of our phototroph, which indicates that the actual presence of the phototroph cells are not required, but it's something that they release in the supernatant that triggers the production of these compounds. And we think that this is actually photosynthate. So it's basically the organic matter that they release when they do photosynthesis. So just as a quick summary of what I said so far, we've shown that Salinispora growing with different marine phototroph release an antimicrobial molecule that is able to kill both eukaryotic and prokaryotic phototrophs. We've shown by LCMS that photosynthate specifically triggers the production of novel cryptic compounds in Salinispora that we've partially characterized. And the next step was to try to identify which gene clusters in Salinispora was responsible for the production of these novel compounds. So to do this, we've used shotgun proteomic to look inside Salinispora cells, and we looked at its proteome. So for example, here you have the 20 biosynthetic gene clusters of Salinispora, where each gene is colored based on the relative abundance of the corresponding protein. So for example, if you see a gene colored in blue, it means we detected a protein in low abundance. if you see a gene in red, it means we detected the protein in high abundance. And when the genes are in black, it means we didn't detect those in our data sets. So the way we've done this is because we wanted to know which gene cluster were responsible for the production of these compounds. And we knew that these compounds were only produced in response to photosynthate or something released by the phototroph. We've looked at the proteome of Salinispora grown exanically in marine broth, where we know they don't produce the compounds, and also in the supernatant of the phototroph, where we know they produce the compound. So for example, here, if you look at the gene clusters that are highlighted in orange, they represent the characterized gene clusters of Salinispora. For example, if you look at the SAL gene cluster that produces salinisporamide, which is again the anti-cancer molecule that I mentioned before, we detected most of the protein from this gene cluster and some with quite high abundance, which makes sense because we also detected a lot of this compound by LCMS. So this is just to show that you can correlate the level of expression of gene clusters seen by proteomic with the actual production of a compound. So what was really interesting about this data set, obviously, was these gene clusters in green, which are the orphan gene clusters, which represent this 60% of potential that I mentioned before. And one gene cluster caught our attention. So this NRPS1 gene cluster here. So we detected quite a lot of protein from this gene cluster. And specifically, we detected this condensation domain containing protein, as well as this non-ribosomal peptide synthetase, which are two enzymes that are key in the biosynthesis of a non-ribosomal peptide molecules. So basically observing these two molecules in our data set strongly suggests that an NRPS molecule is being produced. And we've also detected this MFS transporter, which is sometimes linked to the secretion of antibiotic. So we've then looked at the level of expression of this protein in our different conditions. So here you have the fold change of the detected protein when Salinispora was grown in marine broth or in the supernatant of the phototroph. And if you see a protein in red, it means it was overexpressed in presence of the phototroph. And if it's blue, it means it was underexpressed. And there's one protein in particular that was highly overexpressed, and it was actually this MFS transporter that I just mentioned. So what we think might be happening is that this NRPS1 gene cluster sort of has a basal expression of the molecule it produces. But when the phototroph is here, Salinispora just secretes more of this compound by overexpressing this MFS transporter. And in terms of trying to understand what this molecule could be doing, we've then found this protein that was in our data set, this ATP-dependent click protease. So it was very interesting because virtually all living organisms rely on a proteasome for protein turnover, so to degrade proteins in a specific way. And there are two main types of proteasomes. So there's this ClP type proteasome and also this 20S type proteasome. And there's a class of antibiotic that is known to block this proteasome, which leads to cell death. So it acts as an antibiotic. And it's been shown as well that very often cells that do produce these antibiotics to be resistant to these antibiotics, they will have an extra copy within the gene cluster that produced the antibiotic or nearby that allow them to be resistant to it. And it's actually something that has been shown before in Salinispora itself. So if you look again at this SAL gene cluster, which again produces salinisporamide, which is this anti-cancer molecule, salinisporamide is actually a 20S proteasome inhibitor. And that's this function that allows this molecule to kill cancerous cells. And so Salinispora produces compounds, and to not suffer from its own antibiotic, it's been shown previously that within this gene cluster, there's extra copies of these subunits of the 20S proteasome. And it's been shown that having these extra copies of these subunits of the proteasome allows the cells to be resistant to this molecule. So similarly, if you mirror these to this NRPS1 gene cluster, we think that it produces an NRPS molecule, which probably has a clip, which is maybe a ClP proteasome inhibitor, and that can act as an antibiotic, and that the presence of this ATP-dependent clip protease, this extra copy of this ClP proteasome, allows the cells to be resistant to its own antibiotic. So just to add that in a bit of context, we've shown that Salinispora was able to kill both different type of marine phototroph, and the proteomic allowed us to identify this NRPS1 gene cluster, this orphan gene cluster, as being active. And we think that it produces a ClpP proteasome inhibitor. And we also think that having this extra ClpP copy within the gene cluster allows the cells to be resistant. But in the case of the phototroph that rely also on this ClpP proteasome, we think that this molecule might be the killing agent that kill the phototroph in our co-culture. So the next step in my project was to verify this hypothesis. So to do this, I wanted to engineer the genome of Salinispora by knocking out this NRPS1 gene cluster. So the way I wanted to do this was to use CRISPR-Cas, which has never been used in Salinispora before. So I've used this P-CRISPOmyces plasmid that has been optimized for soil actinomycetes. And I've engineered it to delete different PPTases in Salinispora, as well as entire gene clusters, so the NRPS1 and NRPS2, but the NRPS1 being the one of interest here. So after I engineer these plasmids, I've wanted to conjugate those into Salinispora, which was actually very hard. So I spent 6 to 7 months just trying to make conjugation happen. And I played with a lot of different of these conditions, donor strain, antibiotics, concentration, etc. And it was just, a bunch of empty plates for months until magically one day I've had X-conjugates for a few of my constructs. And I've been able to show, for example, for these PPTAs here by PCR that I was able actually to delete the PPTA, and I've also sequenced the genome to verify that, I indeed deleted the portion of these PPTAs that I wanted, which was very exciting because, again, CRISPR-Cas has never been used in Salinispora before, so that was very exciting for us. Then we moved to trying to knock out entire gene clusters. So we've tried to knock out this NRPS2 gene cluster. And again, by PCR, I've been able to show that one X-conjugate, number 5, was a successful mutant. So we successfully deleted the entire NRPS2 gene cluster. And then I've tried obviously on this NRPS1 gene cluster. And I've tried several times actually a bit before the lockdown, and unfortunately checking, so I did get X-conjugate, but checking them by PCR, they were actually wild type strains. So yeah, it was a bit sad. I didn't manage to delete this NRPS1 gene cluster, but I've been able to show that CRISPR-Cas9 can actually work in Salinispora. It can be used to delete single genes as well as entire gene cluster, which is quite cool. So yeah, this is about the part of the project I wanted to show. So I just want to thank my PIs, Joseph Cristioleza and Chris Core, their groups, my funding, and my VTP, and all of you guys for attending and your attention.
Awesome, thank you for that. So we have a few questions in the chat already. I will just go through these. So Ashok GB says, what class of compounds are these new antimicrobials, which I believe you might have touched on already. But...
I'm sorry, I didn't hear.
Yeah, sorry, they say, what class of compounds are these new antimicrobials?
So they might be proteasome inhibitors.
Okay, cool. So David Marks says, it looks like the PKS3 cluster was also being expressed. What was the reason for focusing on the NRPS1 cluster as opposed?
Yes, that's a very good question. So the PKS3 was also extremely active, but there is literature that suggests that this gene cluster produces a pigment, even though the actual molecule was not identified. So we decided to focus on NRPS1 just because we had an idea of the mechanism of action. Because of this ClpP, we suspected it might be an antibiotic, so we focused on that. But obviously, yeah, the PKS3 is also very interesting. Okay,
cool. Interesting. So Lorena says, really nice talk. Are there any positive regulators in the NRPS1 cluster that you could overexpress to try and increase the yield of the compound?
It's a good question. I've looked at that a while ago, and I don't remember being any type of regulators, but I might, be wrong. But I think there was not.
Okay. And Keaton Nag says, great talk. Are these antibiotics secreted, as you mentioned, about the MFS transporter. Have you tested the antibiotics against any pathogens? So I guess they mean the purified compound.
Yes, so that's a good question, and it's something that we've tried through different means. But the problem is that because I've used a phototroph for microcultures, they're actually very tricky to work with. For example, you cannot plate them as a loan of cells, for example, the phototroph, and also they're quite picky to grow and things like that. So it was quite hard to do this type of assay because they would very often just die because they're picky. So it's hard to have a clear setup. But I did try this kind of stuff, like to, for example, grow Salinispora and then remove the cells and then put Synechococcus into these supernatant. But we didn't observe any death. And I've also tried to spike these different fractions that I collected to see if one of these fractions would have antibiotic effect, but we didn't see anything. But it might be due because the molecules, maybe they're unstable, maybe they're just not enough, maybe there's different molecules that are required for the killing. I mean, it was quite hard to, so the clearest way for us was just to knock out this gene cluster and see what would happen.
Right, yeah, okay. Cool, so we have a question from Kate Duncan. She's asking, if the molecule is a non-ribosomal peptide, can you see the amino acids in MSMS fragmentation data? She says she may have missed it, but did you test your metabolite extract or fractions for the bioactivity as well? So that's the question.
Yeah, so testing the bioactivity, I answered that, I think, before.
Right, yeah.
And so we've tried to do MSMS on this molecule and basically we didn't see any. So what we saw was that there were 4 main fragments by MSMS, which was consistent with potentially a tetrapeptide. But then this difference in mass between these different fragments were not consistent with any known amino acids. So we didn't really know what it could be.
Okay, cool. So we've got time for a couple more questions, I think. So there's a question from Enzo de los Santos. He says, hi, great talk. I may have missed this, but where did you get the genome from? Looking at the gene layout, it might be the NRPS1 cluster might be fragmented. Have you looked into this?
This NRPS1 might be fragmented?
Yeah, that's what they're suggesting, yeah.
Okay, so there's actually one thing that I've noticed when I was studying this gene cluster is that it's very close to another gene cluster, which is PKS1, which is also an orphan gene cluster. They're quite close in the genome. And when you look at the PKS1 gene cluster, or the other way around, they have, for this PKS1 within the gene cluster, there are like genes that may be involved in NRPS biosynthesis. So what I suspect is that these gene clusters are actually maybe one, and they may produce a hybrid polyketide and NRPS. And that would actually make sense because Salinispora, which is this 20S protease inhibitor, is actually a hybrid polyketide NRPS. So maybe this compound is the same, sort of similar, but it's a cryptic protease inhibitor.
Okay. So
I don't know if that answers the question.
Cool. So I think that's probably all the time we have for questions. We should move on to our next speaker, but thanks for that. That was a really cool talk. So our next speaker is, if you could screen share now, is Jonathan Parra, who's based up at Strathclyde.
Okay, thank you everybody. I'm really glad to be here. Thank you for the opportunity to share the work that we are doing with Pseudonocardia strains isolated from the deep Southern Ocean. The first question to ask is why we should study the deep ocean. As probably most of you know, around 70% of the planet's surface is covered by the ocean, and from this water body, almost 50% is considered as a deep ocean. However, the deep ocean represents a big part of the planet. Only few parts of it have been studied yet. This is why it's considered like as the last frontier on Earth. On this scenario, many projects have been related with the exploration of this unknown environment. One of them was the AnteED project, which was a multinational project consisted in 3 different expeditions with the idea of exploring the biodiversity in terms of eukaryotes in the Southern Ocean close to the Antarctica. The Scottish Association of Marine Science, which was part of this project 10 years after, started to be curious about the biodiversity in terms of prokaryotes. Here is where our history started. A former postdoc from our group, Natalie Milan, isolate several rare actinomycetes from some samples collected during the Andeep 2 expedition. One of the most interesting things of this project was a group of 25 Pseudonocardia strains, which according to the 16S RNA gene analysis, 24 of them are distributed into novel clades, which with a low relationship with other previously reported Pseudonocardia strains. That is why we believe that maybe we are working with two potential new species. Another strain was also isolated, close related with Pseudonocardia sediminis, another Pseudonocardia from marine environment. However, I will focus on these two clades. This is how they looks like in the plate. In spite of their genetic correlation, the morphology is quite different. Pseudonocardia petroleophila, a non-marine Pseudonocardia, is the closest type strain. Recently, we found that a strain from the Abyssalis clade have activity against the clinical relevant multi-drug resistant pathogen Candida auris, which increased our interest in these strains. Something really interesting about our strain is the correlation between the taxonomy of them and their geographic origin, in the sense that all of the Abyssalis strains were collected from one spot, while the Oceanic strains were collected from two different collecting points. So we were interested in analyzing how these biogeographic patterns can affect the production of specialized metabolites. This is because the Pseudonocardia is a great model for analyzing this. Thanks to the work of Cameron Curry and Matt Hutchins, now we have a good understanding of the relationship between the ecology and the production of metabolites in this particular strain, which are symbiont of farmer ants. For example, in this paper from the Curry group, it was shown a high diversity in the production of metabolites in different populations of Pseudonocardia collected from relative close areas in Central America. Also, it was shown how, in spite of the low diversity in the core genome, the population in the Barro Colorado island showed a high number of diverse and unique biosynthetic gene clusters, which means a high level of local adaptation to the environment, which probably is related with the geographic isolation of the island. We are looking for find this kind of patterns in our strains, since in the ocean is quite difficult to define the geographic barriers. We believe that if we compare the specialized metabolism of our strains between each other and with other strain isolated from other marine environment, we can have a better idea about the evolution of the specialized metabolism in the genus. In order to do that, we sequence the genome of 8 strains. 2 of them, one of each of the Oceanic and Abyssalis clade, were sequenced using long-read technology and short-read technology. Thanks to this, we obtain a high-quality complete genome of both clades. Then we also sequence the genome of 6 more strains using short-read technology, and we use the complete genomes as a template for scaffolding these genomes. In this way, we obtain a high-quality draft genome of these strains. once that we assemble the genomes, we measure the genomic distance in between our strains and also between our strains and another previously reported type strains. What we found is that the average nucleotide identity between both clades and other previously reported Pseudonocardia strains was less than 80%, and the relationship between our clades have an NI of around 87%. That confirm that we are, at least in genomic level, working with two new species, and these new species are close related to each other. Then we analyze how these genomic differences can affect the specialized metabolism. For this, we annotated the biosynthetic gene cluster, or BGCs, using anti-smash, and we analyze the differences in this biosynthetic gene cluster using the Big Escape platform. What Big Escape does is to cluster the BGCs in gene cluster family according with the similarities in the sequences. Here, what we are seeing is in blue squares the biosynthetic gene cluster, and BCGs in the same column means that they belong to the same gene cluster family. Also, the red square represent BCGs from the MIBIG database, which is a curated database of previously reported biosynthetic gene cluster. What we found is that there are not close related BCGs in the genus or in the MIB database to the BCGs that we annotate in our genomes, which suggests that we are having some kind of novelty in the BCGs of our strains. This is the network output of Big Escape. Here, each node represents one BCG. Two nodes linked means that their sequence are related. What we found is that there are some groups of BCGs that are widely spread in the genus, like terpenes, ectodyne, or bacteriocin, but also we found some group of BCGs that are clade specific. That means that we only found them in one of the clades. Also, we found some groups which have BCGs from both of the clades. When we match this output with the phylogeny of our strains based on the AutoMLST analysis, we obtain this. Here again, each node represents one BCG, and nodes in the same column means that they belong to the same gene cluster family. So we identify one core group of BCGs which contain a biosynthetic gene cluster from the both clades and also from Pseudonocardia hydrocarbonoxidans, which is a close related non-marine Pseudonocardia strains. And also we annotate some group of BCGs, or BGCs, sorry, that are clade specific. That means that they belong only to one of the analyzed species. In order to confirm this, we carried out a preliminary metabolomics analysis. After a year working with these strains, I realized that one of the most challenging parts is to express and extract the metabolites that are being produced by the strains. So after many tries, we found the right condition for doing this. So we grown one strain from each of the clades, as well as two representative Pseudonocardia strain from marine origin. We grow them in solid agar during 28 days using an elicitor. After that, we dry the agar and extract it using ethyl acetate for analyzing by LCMS. Briefly, the spectrometric data was treated using MC-MINE. We removed the noise and aligned the chromatograms in order to obtain matrix data, which was analyzed by multivariable analysis using the metabolomics platform. what we obtain is this heat map. In the columns, we can see the metabolites that are detected for each of the strains by triplicate. What we found is that there is one group of metabolites that are detected in both of the clades or in both of the strains, but also there are one group of metabolites which are detected in only one of the strain, which agrees with the information that we got from the genomic analysis. So in summary, what we are seeing is that there are a core group of biosynthetic gene clusters that is shared in both of the two new species. And also there are a group of biosynthetic gene clusters that were acquired after the speciation process. This situation agrees with the ecology theory of the evolution in BGCs that said that when one microorganism is dispersed into different niches, some of the biosynthetic gene clusters that are not essential for the adaptation to this new environment are swept from the genome, while another gene cluster acquired via horizontal gene transfer, and the biosynthetic gene cluster, the new biosynthetic gene clusters that are important for the survival in this new environment, are integrated in the genome and passed to the next generation, generating a diversity in the specialized metabolism during the speciation process. In order to analyze the role of the horizontal gene transfer in the evolution of the biosynthetic gene cluster from our strains, we annotate genomic islands in our genome, and we found that there are some group of biosynthetic gene clusters that are flanked or integrate into genomic islands. We found two different situations. In one of them, we found some biosynthetic gene cluster related with genomic island that are part of gene cluster families. So here what we propose is that the horizontal gene transfer affect the evolution of this biosynthetic gene cluster, generating changes in the structure of the BCG. And also we found some BGCs with evidence of horizontal gene transfer that are clade exclusive. In this case, we have two possible scenarios. In one of them, the whole biosynthetic gene cluster was transferred horizontally to the genome. And the another scenario that is the more probable, in my opinion, is that we are here in front of the same situation that I just explained, where only some genes were transferred horizontally, modifying the structure of the biosynthetic gene cluster, but these BGCs were swept from the another clade. So in this way, I can propose some theory where one common ancestor had the core BGCs, and during the dispersal process to the marine environment, some of the biosynthetic gene clusters that were not essential for the adaptation to this new environment were swept from the genome, while other ones were acquired horizontally, generating the diversity in biosynthetic gene cluster that we are seeing now. The next step of this project is to scale up the metabolomics analysis, increasing the number of analyzed strains, and also use LC-MS data in order to work with the molecular networking approach. After that, the idea is try to link the genomic information with the metabolomic information in order to have a big picture of the complete metabolism of our strains. In summary, I may say that around 50% of the analyzed BGCs are clade specific and show us some evidence of horizontal gene transfer. That means that the dispersal process and the speciation process drop the diversity in the specialized metabolism that we are seeing in our strains, and we confirm that the new species that we are obtaining from the Southern Ocean are a potential source of novel antimicrobial. To finish, I just want to say thank you to the Ministry of Science and Technology of Costa Rica and the University of Costa Rica, who paid my fellowship, also to my supervisor, Catherine Duncan, to the Professor Carol Murrow from the University of Aberdeen, who helped us with the antifungal test, and also to the Dr. Natalie Milan from the Universidad Autónoma de Baja California, and of course to my group, Duncan Group, and the World Microbiology Group in Strathclyde. Also, I just want to say thank you to the ActinoBase and the organizers, Sam, David, Alicia, Tom, Emily. Thank you so much. And that was me. Thank you so much.
Awesome, thank you for that. That was a really nice talk. We have a few questions already. So there's a question from Satria Kortsa, who says, can you provide more details about the sequencing strategy used for PacBio and Illumina? And they're asking about how many raw reads per sequencer, and it resulted in a single contig. So I guess a technical question.
But what is the question in specific about what?
So they're asking for more details about the sequencing strategy you used for PacBio and Illumina. How many raw reads per sequencer, and was there a single complete contig, or was it multiple?
Well, for the complete genome, we obtain the whole chromosome in, let me show you, in one contig, and in both of the cases, also in Avisalis and Oceani, we obtain one big chromosome and one plasmid. It was possible to see this only in the chromosome, in the assemblies, using the long reads. For the others, the contiguity was low. After the scaffolding process, we also obtain a few number of contigs, around 10, depends of the coverage of each of the genomes, but the number of contigs in all of the cases was less than 10.
Cool, awesome. So there's the same question here from two people. They're both just asking what the elicitor you used was for the metabolomics analysis.
We tried 3 different elicitors, and we obtained positive result with 2 of them. N-acetylglucosamine, which is a well-known elicitor for Streptomyces, and also N-acetyl. Well, it's another butyrolactone, I think. Yeah, sorry, I forgot specific the name. But it's an elicitor that it was used before in the group of Matt Hoshin and Cameron Curry for eliciting Pseudonocardias from left cutter ants.
Right.
The information that I'm showing, it was using N-acetylglucosamine, which triggered the production of metabolites in more of the strains. Some of the elicitor was better for some of the strains, so we choose the one that produced more diversity in most of the strains.
Cool, awesome. So there's a question here. Nice talk. What percentage of genes were transferred? Any specific function of the genes transferred? I presume I mean across strains.
Well, in some cases, it was reported before for Pseudonocardia from ants that the whole gene, biosynthetic gene cluster are transferred between a strain using plasmid. We couldn't find biosynthetic gene cluster into plasmid in our strains. What we found was evidence of horizontal gene transfer next to the biosynthetic gene cluster. So we are not proposing the horizontal transfer of the whole biosynthetic gene cluster. We are talking more about some kind of genomic recombination between the both kind of clades, which generate the diversity that we are seeing now.
Cool, awesome, okay. Yeah, I guess that Rebecca also asked a similar question Do you have any idea which horizontal gene transfer methods resulted in the genomic islands? Well, I guess you kind of already answered that. Let me just have a look. So there's a question here. Is it possible that the template scaffolding of the other draft genomes biased their phylogenetic placement?
Sorry, say again?
So the question is it possible that the template scaffolding of the other draft genomes biased their phylogenetic placement at all?
Well, yes, it is possible in the sense that you are scaffolding one of the strains using the another strain. However, we okay, let me show you this. We use different ways to analyze the phylogeny of the strains. For example, using the core genome, also running the autoMLST analysis without scaffolding process, and using the 16S analysis. And in all of the cases, the phylogeny was exactly the same.
Right. Cool. So I think that seems to be all I can spot questions-wise. So thanks for that. Thanks for the questions, everyone. And thanks to both of our speakers for some great talks today. Give me one moment. I will just share my screen. So for next week, we have we'll be back next week, sorry, at 2 o'clock, so a bit earlier from the UK perspective anyway. And we will have Bernhard Keplinger from the University of Newcastle and Patrick Capel from University of Warwick speaking. So yeah, thanks for dropping in today and for the great questions, and thanks to both our speakers today. And we will see you all next week. Thank you.